Welcome back to Biology with Zimmerman. Today we're going to be looking at two inheritance patterns, incomplete and codominance. These two can get confusing because what we see in them is very similar in the fact that the alleles that code for these traits, neither is really dominant and neither is really recessive. So if we look at this idea of codominance, both of those alleles are expressed equally. So in this particular flower here, you have a allele that codes for red and you have an allele that codes for white. When you put those together, you see red and white in the phenotype. We see this kind of inheritance pattern with roan cattle. Think Texas Longhorn cattle. You can have roan cattle, which are red and white spotted, or you could have white cattle, or you could have red cattle. So when we are trying to figure out alleles for these, you can choose whatever letter you like. Sometimes you might see red represented with a capital R and white represented with a lowercase r. I don't particularly like doing it that way because it makes me think that white is recessive and it's not. So I'm going to pick a big R to represent red and I'm going to pick a big W to represent white. So a red flower is going to have the genotype big R, because you need two copies of that. You have two copies of that. White is going to be WW, and this spotted flower is going to be RW, or you could write it as WR. It really doesn't matter. Now with incomplete dominance, you don't have this equal expression in the heterozygous state. The alleles blend together. So think about what happens when you mix red and white paint together. You get this pink coloration pattern. So red and white coming together is going to give you pink. We do the alleles the exact same way. So red would be an R and white would be a W. So red would be RR, W would be, or white would be WW, and pink would be RW or WR. So when you're doing these kinds of problems, it's just really important that you pay attention to this heterozygous state. If they say, when you put these two alleles together, you have spots or you see both of them, that's going to tell you it's codominance. If they put those two different alleles or two different traits together and it mixes, then it's going to be incomplete dominance. The way that you do these genetic crosses is exactly the same. So we're going to do an example crossing a red with a spotted. So red, we said our genotype is big R, spotted we said is RW. So I'm going to show that cross in a Punnett square. The Punnett square is done exactly the same way as we've done all of our genetic crosses. So you're going to fill across and down, across and down. And what we end up is a Punnett square that looks like this. So our genotype percentages, we have two boxes that are big R, so that's going to be 50% are big R. Two boxes are big RW, so that's going to be 50%. And if we look at the phenotypes, that's going to correspond to 50% being red and 50% being spotted. Now I can switch this up, and if this was an incomplete dominance problem, instead of saying that I crossed it with a spotted, I could say that I crossed it with a pink. So we're going to keep that heterozygous genotype the same. All of this would be the same. Now we're just taking that spotted heterozygous phenotype and changing it to a pink. So you do these problems the exact same way. You just have to pay attention to what's going on in that heterozygous state. There aren't too many genetic traits that are inherited this way. In humans, you will see one example, and that's with our blood type. Now, blood type gets a little bit confusing because not only is it codominant, it also has multiple alleles. So instead of having 2 alleles for a particular trait, our blood types have 3 alleles. So you could have the A allele, you could have the B allele, or you could have the O allele. And it is codominant because A and B are codominant to each other, and they're completely dominant over O. So A and B are codominant, and they are completely dominant over O. So O is recessive. So if we look over here at this blood type chart, you could have 3 different phenotypes. You could be, 4 different phenotypes. You could be A type blood, you could be AB, you could be B type blood, or you could be O type blood. So if we look back at the genotypes, for A type blood, you could be homozygous for A, you could be AA, or you could be heterozygous, and we represent that with an A and an O. Remember, O is completely recessive to A. So when you put those together, we're going to see A. You could have AB type blood, and that genotype is going to be AB. They're codominant, so we're going to see both in the phenotype. For B type blood, you could be homozygous for B, so 2 copies of that B allele, or you could be heterozygous. And remember, O is completely recessive to B, so we're going to see B in the phenotype. And then you can also have 2 O alleles and be O type blood. So for me, my blood type is type B. And I know that my mom is AB blood and my dad has O type blood. So figuring out my genotype is going to be pretty easy. My mom's genotype is AB, and my dad, since he has type O blood, he's going to be OO. So if we were going to do a genetic cross looking at my parents, ooh, that's crazy. Punnett squares are filled out the exact same way. I don't know why this is happening. We're going to go with it. So that's what my genetic cross would look like. So when my parents had me, there was a 50% chance they'd have a child with B-type blood and a 50% chance they would have a child with A-type blood. So this is really useful for different sorts of things. In the past, they would use blood typing for paternity test. Before DNA testing, they would use it for matching things for crime scenes. So if you come across a genetic cross with blood typing, you just need to pay attention to the phenotype that they have and what their potential genotypes could be. All right.